Thank you very much for inviting me to present in person this year. I'm very glad to be here. So today I'm going to give an introduction about what we're doing in the macroevolution unit at the Okinawa Institute of Science and Technology, which has been established for about 2 years now. So macroevolution is the study of biodiversity processes above the population or ecosystem level and beyond human generational timescales. So at a much sort of higher spatial scales and higher temporal scales than what's been covered in a lot of the other talks during this symposium. So in terms of macroevolution, what we're interested in the unit is things like trait change, both over time and across ecosystems, features like extinction, both mass extinction, local extinction, species extinction, diversification, how we get so many species and why some thrive and some fail, and then also factors like ecological change. So what drives ecological changes? Do ecosystems change abruptly? When do they collapse? How do communities come together and form new ecosystems? So of course, because I'm speaking in the symposium, obviously we use fishes as our primary study group because they're species and key members of aquatic ecosystems. So I don't have to explain that too much here. Modern fishes are of course diverse and they show a lot of different shapes and a lot of different traits. There's a lot of convergent evolution among them. And if we look back at extinct fishes through their 500 million year fossil record, we find that extinct fishes, that is any vertebrate that does not have a terrestrial ancestor, were even more diverse than what we see today in terms of their lineage history, in terms of their ecology, in terms of even features that we don't necessarily see among the modern fishes. But of course, a lot of those early fishes were also convergent on modern fishes. And so what we have when we look across both the living and the fossil record of fishes is a number of natural experiments in time and space that tell us something about the factors that drive the structure, that determine the structure of biodiversity, both at local scales and at global scales. So for example, we have, say, multiple mass extinctions that have happened in the history of life on Earth. Each one of these has reset the clock in terms of diversification or biodiversity structure by eliminating whole biomes, for example, or whole groups. And so we can see the processes of diversification play out after each one of these events. We also have things like archipelagos or islands, which of course in island biogeography we can use as microcosms to see how ecosystems are assembled, how they're populated, and then compare similar islands in different places. So the first major effort that we take in the unit in terms of looking at biodiversity is reconstructing past biodiversity from fossils. So I'm a paleontologist by training, although I was originally a biologist. I don't make a lot of distinctions between dead and living things. But basically, in order to reconstruct past biodiversity, we start with the fossils themselves. And unlike what you may see in movies or in museums, most fossils are actually absolutely terrible, especially fish fossils. So I showed the nice ones on a different slide. A lot of them are broken up like this. And so the first effort that we have to go through is reconstructing these fossils into something approximating what they looked like in life. And so that's how we end up in the drawings in the second panel. We can assemble all of the species that are described just like any other species into a faunal list, so we know everything that lived around the same time in the same habitat. And then we can look at changes in biodiversity structure through time and changes in ecosystems through time and apply the same kind of ecological statistics and ecological comparisons that you would use in living things. However, there is a change in scale. So we usually look at species, say, within groups when we make these comparisons. We don't have a lot of abundance data for most of the species. And that's just because of the way that fossils are collected for fishes, where the focus has been on really interesting animals, or maybe someone finds these fish while they're looking at a more interesting dinosaur and it gets thrown in a drawer for 200 years. And so we don't have an idea of how many were originally there, but we're trying to address that as well through new field work. So by looking at all of this, we can keep track of changes in global biodiversity through time. Things like shifts in habitats, changes in assembly, when some species originated, when some species went extinct, and why that happens. So I'll just give some examples of the kind of results that we've gotten from these efforts. So first is we can look at mass extinction and turnover under similar conditions. So here's two mass extinctions. One is one of the big 5 mass extinctions, the Endordovician mass extinction from 443 million years ago, and this is new data. And the other one is the Devonian mass extinction from 359 million years ago. So in one mass extinction, there were things that we may attribute to the mass extinction itself that are not necessarily the result of that, or just specific to one group with nothing that can be inferred for larger groups. In this case, we have two extinctions that are driven both by climate changes. First, rapid warming, and then rapid cooling, including the first appearance of polar ice caps in several million years. And so by looking at fishes, what we can see is that in each case, we have extinction in the group of fishes that were dominant before the mass extinction, at lower numbers before the end Ordovician, because fishes with bones had just appeared. And then we have this gap in the fossil record that has been named from maybe several decades ago. Gap meaning that there's low numbers of species when we find them, there aren't a lot of fossils, and this is the initial recovery period. And then we have this large diversification of the fishes that come to dominate the next interval. And we can look at whether this replacement was associated with rapid or gradual turnover by looking at histograms or doing other analyses of the ecosystems themselves and seeing a change in what species and what groups actually dominate those ecosystems. And so in each case, we find a changeover from, again, the fauna that was dominant before the mass extinction to a fauna that looks very different afterwards. So you don't need to pay attention to the groups too much, but just look at the color changes, which represent major groups. And by major groups, I mean actinopterygians versus elasmobranchs level of groups. We can also visualize these through ordination analyses, which tell us exactly what species are driving the differences in ecosystems before and after the mass extinction and how distinct they are as well. So we can do statistics on this. So at the end Ordovician mass extinction, we see a split between this triangle down here, which represents all the pre-extinction ecosystems, and then all of the ecosystems that come after, and then the same thing over the end Devonian mass extinction. So we have repeated extinction processes and recovery processes that are creating the same patterns in two very different time intervals based on the same triggers. After mass extinction, we can also look at downstream or related effects on ecologically connected groups. So in the case of the end Ordovician extinction, this group called conodonts, which were actually cyclostomes, had been dominant in reef settings and in all deeper waters before the mass extinction. Their average diversity goes from about 70 genera worldwide to about 10 to 20. And then afterwards, you get the diversification of free-swimming reef-associated fishes for the first time, fishes related to jawfishes, I mean. And then the same thing after the end Devonian extinction, you have the extinction especially of shell-crushing fishes in the event itself, followed by the diversification of crinoids, which are echinoderms, followed by the new evolution of new crinoid predators. We can also track regional loss and recovery, how biogeography affects these recovery patterns and diversification. So in this case, we can see that after the end Ordovician mass extinction, that while we had fishes present in what's called Gondwana, which is all the southern continents together, that after the mass extinction, during the mass extinction, they were extirpated entirely, and we have no fish diversity after that point. We can also look at patterns like that jawfishes, which show up in the record for the first time after this mass extinction, only appear in one isolated spot in what is now South China, separated by a deep ocean from basically everything else. We can also track habitat dispersal in these new clades as they're diversifying. When do they get to fresh water? When do they get to deeper water? We can track invasion and turnover, in this case between jawed and jawless fishes, that even though they both originate after the mass extinction, as I said, the jawed fishes are initially restricted to, in purple, this South China area. And then by looking at the composition of ecosystems, we can see this invasion wave spread out, where they start to dominate ecosystems spreading out from their point of origin, which confirms that that record where they're restricted South China is probably a real record. However, while we can do all these things, we do have gaps, of course, in the fossil record, where we need data from living animals in order to test our hypotheses, in order to make sure their inferences from the record of biodiversity are true. So we need to be able to infer missing structures like soft tissue so that we can make comparisons for convergence. We can infer missing ancestral states from trait distributions and models. We can infer evolutionary mode for diversification from models. We can infer the ecological meaning of traits from comparisons with living fishes. We can infer function from biomechanical tests. And we can infer growth from fossil series of different sizes and from models of the same. We can also look at changes in traits once we have the species themselves, so we can measure things like body size, and then see how body size changes on a global level in relationship to sort of global environmental factors, but also in terms of competition and other things. We can also track ecomorphological changes by looking, using analyses like geometric morphometrics to capture shape, and then relating this to what we know from modern fishes in terms of biomechanics to see how the distribution of shapes changes over time, especially after the extinction. However, the problem with geometric morphometrics is that you need homologous structures. So more recently, what we've started to do is use ecomorphologically meaningful variations instead that can be applied to a number of different fishes, trying to create a morphospace that is a space that maps out shape for all fishes through time. So here we have data from early fishes from the Silurian, Devonian, Carboniferous. So that's from about 440 million years ago to 320 million years ago. And then we have, from FishBase data, the same coding applied to the members of large ecosystems from Australia, the Northeast US, and the Canadian Arctic that shows that they all fill about the same space with different clustering, and that space was filled gradually during the Paleozoic and has only been replaced since then. And then, of course, using this, we can see turnover in ecology in, say, the jawed versus the jawless fishes. However, while we have all of this, we need more living data to make better inferences, which is why I'm glad to be here. So we need traits in ecology to be able to infer function. We want to know which features of modern ecosystems result from shared processes, so we can infer that as well. And we want to know how local changes scale up and how predictable they are. So I know while I'm running out of time a little bit, I want to talk about the other thing that our lab has started doing because I'm in Okinawa. So this is a new effort in terms of my own research, which is using Okinawa as a missing piece in global databases. So a lot of the fishes there aren't cataloged, for example, and it's thought that 50% of the species aren't actually described, but are known to the fishermen. And as a model system in terms of having an island chain that's continental that is affected by sort of large-scale environmental changes and ocean currents and things like that, to see how communities are assembled over time. Okinawa is also a good model system because it has suffered multiple disruptions at different scales. So as I mentioned, it's continental, so going back only sort of over the last 1.5 million years, at several points it's been connected to the mainland, either of Japan or of Asia as a whole. It also undergoes periodic disturbances, such as typhoons and tsunami, and then it suffers from stochastic events. Most specifically, around 80 years ago, during the Battle of Okinawa, all the reefs were destroyed. However, despite this history of disruption, looking at the global data that are available, we see that the island chain actually has some of the highest biodiversity recorded in Japan, even though, again, a lot of the records are missing, and that there is differentiation between the different islands in terms of their communities already. So in order to bulk up this data and really look at what's happening, we are building a fish collection from different fish markets that are very limited in terms of where they collect from. So each fishing corporation has access to one small area, and they guard it very jealously, and they fish mostly by small boat or by spearfishing, so we know where most of these fish came from. Cataloging their morphology, dissecting them for specific traits so that we can get very high detailed data on some traits that may not have been accessible before, like cells or gill rakers and things like that. We've also most recently started fieldwork surveys to look at diversity in the wild using GoPros and then leaving them in various transects for an hour across the island, and then going through and coding the data for behavior, for what fish species are there, what time of day, what the temperature was as well. And then we've also connected to people capturing larger survey data in the Indo-Pacific out of scripts to look at how much endemism there might be in these different islands and how the communities compare to each other. So the aim of this is to determine the influence of these environmental factors and events and take even genomic data to look at population connectivity across the islands, and then also to look at potential genetic bottlenecks within the species themselves to see if there is an effect of these past events on their diversification or their numbers or their distributions afterwards. So basically, the goal of the unit is to reconstruct and understand fish biodiversity patterns and processes against space and time. And so with that, I'd like to acknowledge the unit members that are leading the various projects that I tried to summarize in 14 minutes, which was difficult. So some of these are interns like Wahe and Rika. Some of them are PhD students like Nanako, and then some of them are postdocs like Sam Kotham. or Chloe Nash, who is leading our fieldwork efforts and has worked in French Polynesia before, or Tai Kubo, who has now moved to a faculty position at Waseda University, and then, of course, other co-authors on these projects. So let me know if you're interested in Okinawan fishes or discussing the diversity there or anything else, and thank you very much for listening.